So thanks everyone for turning up for today's talk. Before we get started, of course, I want to acknowledge the traditional custodians of the land. For many of us, that will be the Wurundjeri people of the Kulin Nation, and where we are located, wherever we are located. I'd also like to pay respect to elders both past and present, and extend that respect to other Indigenous Australians present online. And our upcoming events are listed here. I think, again, we've got a busy program, and encourage you all to come along and listen to that. And I think, again, this is one of the upsides of COVID, is that we can now fit listening into some of these great talks, even in our very busy schedules. So if you have a question, I can't quite see that slide, but if you can, there's a Q&A button, I believe, which you can use. And there is also a chat button where you can place your questions. And if you really have problems using either of those two, then you can try raising your hands. Okay, so we're on to the main business of the afternoon. And it's a real great pleasure for me to introduce Simon. He's Simon Chu. He's a research group head of the Hormone Cancer Therapeutics Laboratory at the Hudson Institute in Melbourne. He completed his PhD in Monash and did postdoc work at Prince Henry's in the Pasteur Institute before starting his own independent research in 2009. And as you can see from his title today, his work focuses on ovarian granulosa cell tumors. And I should confess here that I've collaborated with Simon, and it's always been a great pleasure to work together with him on this very a tumor type that really still is not as investigated as it should be. And Simon's going to change all that, and we're going to understand better ways to treat. And I think one of the great wins that he just had recently, and a really great measure of the sort of importance of his work, is that he just secured and led an MRFF bid for priorities in ovarian cancer research. And seeing as you'd much rather listen to Simon than myself, I'm going to stop now and really hand it over to Simon. So thank you very much, Simon, and I'm really looking forward to your talk.
Thanks very much, John, for that introduction, and I'll just put my share screen on here with my presentation now. So, yeah, I am a research group head at the Hudson Institute of Medical Research, and my interest is on these uncommon types of ovarian cancers. So as a bit of an overview, this is what I'll be talking about this afternoon. These particular subjects from the background, a bit of background, some of the markers for this tumour type, and a little bit on the areas of research that have been conducted in my lab, and then also just a little bit about that MRFF grant that we were recently awarded. So ovarian cancers are generally classified into three separate categories. By far the most common type of ovarian cancer are those of epithelial in origin, and by far the most serious and dangerous of these are the serious, high-grade serous ovarian cancers, which are in general what we hear mostly about. But then we do also have more uncommon types of ovarian cancers, and those that are the most uncommon are the germ cell tumours, which account for about 1 to 3%. But then there are also these types of tumours which arise from the sex-cord stromal region, and which are really truly ovarian cancers because they come from within. And we've been interested over many years in this particular tumour type, the granulosa cell tumour, which is by far accounts for the majority of these sex-cord stromal tumours. So these are types of tumours here, these sex-cord stromal tumours, as you can see in this list here, encompass a whole range of different tumour types, but 90% of them are of granulosa cell tumours. And in fact, granulosa cell tumours can be further categorised into two types. Those of the adult GCT lineage, which account for about 95% of all GCTs, which I'll call them from now on. And then we also have a much rarer form known as the juvenile GCT, which is about 5%. In the past, classification of these tumours was primarily morphological, and as you can see in this histological pictogram here, that this is a typical representation of a granulosa cell tumour under the microscope. On the left-hand side is a primary tumour, and it's typically this is a very unique feature of these tumours, whereby these exner cell core bodies, which form what look like very primordial type follicles, appear, and this very distinctive nuclei pattern. But on the right-hand side is a recurrent tumour, which still contains a similar type of nuclear structure there, but this is a recurrent tumour from the same patient. These are hormonally active tumours, and so they actually resemble much like an FSH, a follicle stimulating hormone primed late pre-ovulatory granulosa cell. And I'll explain a little bit about where these cells are located in a minute. But one of the things that we know about these tumours is that they, being hormonally active, they secrete a range of different gonadal hormones and peptides. And some of these are inhibin, anti-Müllerian hormone, and estrogen. And in fact, if you look at this pictogram here, we see a staining of an immunohistochemical staining of a granulosa cell tumour for inhibin, and we know that these are quite hard, or inhibin is overexpressed in these tumours. So this was from a study, we know this from studies conducted over 2 decades ago, whereby elevated inhibin has been demonstrated for those patients who have a granulosa cell tumour. And this is the staining from a pre-operation levels of inhibin, and then post-operation those levels fall to near undetectable levels. It's also been shown to be a very good monitoring biomarker, considering that one can then be able to monitor for the increase in inhibin over time to be able to predict recurrence of that tumour. And so in this graph here, as we see, that post-op, their inhibin levels fall below detection rate, but then sometime post-surgery, the inhibin levels again start to rise, indicating a recurrence. Now, out of these studies that were conducted here in Melbourne across different institutions, it led to our institute being able to collate one of the world's largest granulosa cell tumour bank, and so that we currently hold this at our institute. But it also led to clinical assays for determination of total inhibin, or the bioactive inhibins, inhibin A and inhibin B. These are now mostly in clinical use. However, we know that one of these assays, the total inhibin assay, which is actually the best inhibin assay in order to be able to monitor for ovarian cancer, and in particular granulosa cell tumours, that there's a real clinical need for a better assay than the one that's currently, we don't currently have anymore in this country. Now, some of the clinical manifestations of granulosa cell tumours, the peak incidence is between 50 to 60 years. Mind you, I mentioned that there's a juvenile form of this disease, which accounts for 5%, and the youngest patient that we know of is about 11 months. It can range all the way to much older for that particular subtype. Some of the symptoms are, as I mentioned, these are hormonal type tumours, and some of the symptoms are a result of the excess estrogen secretion. And so, in fact, many patients present early due to these this clinical manifestation. And so some of the associated symptoms are vaginal bleeding or abdominal pelvic pain, and that's to do with the estrogen secretion. And because of the fact that many women are often caught earlier at stage 1 or stage 2 for them for these tumours, because of this excess estrogen secretion, then they're often being able to be treated much earlier. And so hence they generally have a better prognosis than women who have the epithelial type tumours. However, in saying that, approximately 80% of women who have advanced or who get recurrent disease will actually die of their disease. Now, the primary treatment is surgical, but for those with advanced disease, intra-abdominal spread makes complete removal difficult, and so hence many of these women have to go back for multiple operations in order to be able to remove their tumour. These tumours are also characterised by their tendency for late recurrence, and so sometimes even up to 30 years post initial diagnosis has been reported, and so it's a unique feature of this tumour type. And unfortunately, recurrent disease is treated with chemotherapy or radiotherapy and even hormonal therapy, but with really with limited success. And so there's a better need for treatments for this particular tumour type. And the reason for this in many ways is that the regimens that are used, the chemotherapeutic regimens, for example, are often based on the platinum-containing protocols that are used for the epithelial ovarian cancers, which are clearly of a different etiology. So granulosa cell tumours arise from ovarian granulosa cells. So these cells are actually the cells that surround the developing oocyte in ovarian follicles. And a feature of this particular pathway, if you like, or the flow of follicular genesis, whereby the oocyte is nurtured to ovulation, is that under the influence of FSH, estrogen, and other growth factors, there's this rapid proliferation of granulosa cells that then nurture the egg toward the ovulation. Once the egg is ovulated, then these granulosa cells will terminally differentiate into what we know as what we call luteal cells. And then these luteal cells form the corpus luteum, which then is responsible for producing other hormones such as progesterone in order to then support fertilization and implantation and so forth. A lot of our work from the past has been focused on trying to work out the molecular phenotype of these tumor cells, and we've determined that in fact they share a very similar molecular phenotype to cells of a late pre-ovulatory follicle. So somewhere just before that ovulatory phase, where they're susceptible, where the cells are sensitive to the other important gonadotropin luteinizing hormone, these cells don't luteinize at all, but rather they continue to proliferate. However, over a long period of time, the understanding of what causes a granulosa cell to become a granulosa cell tumour, that there was a big black box where we really didn't understand what the molecular events that led to this. And then about just over a decade ago, a seminal paper by Shah et al. In David Huntsman's lab in Canada observed a mutation in the FOXL2 gene, which is responsible for granulosa cell tumours. And this study actually was just a whole transcriptome sequencing, RNA-seq of only 4 granulosa cell tumours at the time, where they identified this one nucleotide change in the FOXL2 gene. They then subsequently looked at some archival material whereby they actually observed this mutation in 86 of 89 adult granulosa cell tumours. Now I'll stress that these are in adult granulosa cell tumours and not any other tumour type. So no other tumours, including juvenile GCT, have this tumour, have this mutation. We ourselves in our unit, in collaboration with a Helsinki group, looked at this mutation in 56 adult GCT and 6 juvenile GCT. And again, we observed the same thing where none of the juvenile GCTs have this mutation, and 52 of the 56 adult GCTs all contain that FOXL2 C134W mutation. When we further looked at the 4 mutation negative granulosa cell tumours, on pathology review, in fact, we determined that 3 of them were misdiagnosed as adult GCT. And so hence it led to us to conclude that this mutation is pathognomonic for adult GCT. We also looked at another study in collaboration with Colin Stewart in University of Western Australia, whereby we looked at the analysis of problematic or unclassified ovarian sex-caused stromal tumours, and we found that 4 of the 11 tumours with satisfactory genome analysis actually showed a FOXL2 mutation, so we could determine that they were adult GCT. 3 of the 4 cases were reported initially as unclassified sex-caused stromal tumours, and 1 as a Sartori-Leydig cell tumour. But then those, conversely, then 3 cases with an original diagnosis of an adult granulosa cell tumour were actually mutation negative, and so we have some doubt as to whether they are really an adult GCT. Now in the laboratory, we have access to just a couple of cell lines for a lot of our in vitro work, and it's an issue that we have in being able to investigate these rarer types of ovarian cancers. And at the time we had access to this cell line called 434 cells, which were derived from a 27-year-old lady who had metastatic cancer. And then a bit later on, access to the KGN cell line, which was isolated from a 73-year-old and was a stage 3 recurrent metastatic tumour. Upon doing the FOXL2 mutation status, we actually observed that the COV434 cells are wild type for the FOXL2 mutation. In fact, there's no expression of this gene at all. And so in the end, we've determined that that's likely to be a juvenile GCT. However, there's some conjecture at the moment as to whether that is truly the case, because there's another group that's found another mutation which might suggest it might be another cell type. However, the KGN cells, which we use as our mainstay in the laboratory, do harbour the FOXL2 C134W mutation, so clearly an adult granulosa cell tumour. And I'll move into a bit later on about how we've looked at the pathways associated with these tumours and see constitutive activity of, there's a couple of common pathways that are constitutively active in both of these tumour cell lines. So we've been interested in trying to understand, at least at the very start for adult GCT, what are the differences between a relatively benign or indolent, slow-growing tumour that gets caught at stage 1, but what causes them to become an advanced or recurrent tumour down the track and relatively untreatable. We know an early event is this acquisition of the FOXL2 mutation, but what are the subsequent events that will lead that tumour to become a recurrent or aggressive? So the questions we've asked are, can we identify markers that will predict recurrence, that will also solve the etiology of aggressive and or recurrent disease, and also potentially whether there are targetable disease-specific therapeutics that we can develop in order to be able to treat this disease better. So our focus from our laboratory has been to look at nuclear receptors, and I hope to be able to show you why in a little while, but also focusing on this particular pathway, the NF-kappa B and the X-linked inhibitor of apoptosis proteins, which we've had a real interest in. We've done quite a bit of work looking at tyrosine kinase inhibitors, but I won't talk to you about that today, as well as some whole transcriptome analysis and whole exome analyses. So nuclear receptors have an established role in granulosa cell biology, and so we've been really interested as a lab that's been interested in steroid hormone receptors traditionally, to be able to observe or look at whether we can develop some type of hormonal therapy to treat this disease, akin to perhaps the anti-estrogens being used for breast cancer. So we focused on a couple of nuclear receptors, which I'll just pull out here for now. The ER beta, the estrogen receptor beta isoform, and PPAR gamma, peroxisome proliferator activated receptor gamma. And I'll go into why we've looked at these in a minute. So estrogen receptor beta is the predominant estrogen receptor isoform in granulosa cells of the ovary. However, the role of this receptor, even though it's been now discovered close to 2 decades, its actual role in granulosa cell proliferation and differentiation is still relatively poorly understood. There is evidence that it's involved in terminal differentiation, but this is a conundrum because we're trying to understand how a receptor that's so overexpressed in granulosa cell tumours could well be or could be involved in this particular pathway. As I mentioned just then, is that this receptor is overexpressed in GCT, and it's also expressed in the 2 cell lines, COL434 and KGN, and hence why we still believe that the COL434 are a granulosa cell derived tumour line. However, it's probably from the juvenile lineage. We were interested in this because one of the types of treatments that has been investigated and is being used in clinical trials as well is the use of aromatase inhibitors, and which has shown there have been some good reports, and then there have also been several which have shown that they're not really that efficacious for treatment of this type of disease. And so the idea would be then that perhaps estrogen would be playing a role in driving this tumor type, and I guess which receptor would it be driving through. But when we looked at the expression of ER beta in these GCT derived cell lines, what we have actually observed was, in fact, the endogenous receptor, when you transfect these cells with a reporter construct that's sensitive to estrogen, that when you treat these cells with a physiological dose of estradiol or the synthetic estrogen diethylstilbestrol, that you don't actually induce this reporter. Now, we know that even if we transfected the cells with an exogenous ER beta or even ER alpha, that we got a very similar response, that these cells were not able to be transactivated and induced by estrogens. So at the time when we're trying to work out why that was, we looked at other signaling pathways as well. And 2 pathways that came of interest were the Ap1 signaling pathway, whereby if you transfected the cells with an Ap1 transactivation reporter, that under basal conditions, as you see here in white for both KGN and COL434 cells, that you could induce the reporter 4 to 5-fold under basal conditions, and which couldn't be further stimulated. So leading us to conclude that Ap1 was constitutively active. That's in comparison to the reporterless construct. But we also observed a very similar thing with the NF-kappa B signaling pathway, whereby transfecting in an NF-kappa B loop plasmid into these cells, that we got, again, a 5-fold basal induction of the reporter compared to the empty vector. But in this instance, that we could further induce this pathway using an exogenous factor, TNF-alpha. So that led us to conclude then that constitutive Ap1 activity and NF-kappa B activity are present in both of these cell lines. So these transcription factors are known to interact with both ER alpha and the glucocorticoid receptor to mutually inhibit transcription. So in the process we call transrepression. And so we asked the question, does transrepression then contribute to the lack of the response that we saw in these cells? And so we know just a little bit about, we focused on NF-kappa B, and the signaling pathways that are involved in its activation are really important for tumour development. And constitutive activation of NF-kappa B is actually an emerging, or is a hallmark of various solid tumours, including, as you see in this list here, breast, colon, prostate, and epithelial ovarian cancers. And in our studies, we actually showed that if you inhibit NF-kappa B with this irreversible inhibitor of that NF-kappa B signaling pathway, BA11-7082, that we're able to induce apoptosis and growth arrest in both of these cell lines. So we asked the question then whether or not transrepression was occurring in these cells. And so when, this is what I showed you just before, this same graph here, but when we introduced a non-toxic dose of the BA11-7082, the NF-kappa B inhibitor, what we observed was, in fact, we were able to induce, upon estrogen stimulation in combination with the BA compound, we're able to reinstall the signaling, estrogen signaling in these cells. And we know that this was an estrogen receptor mediated effect, because when we added a tenfold amount of the anti-estrogen in this as well, that we're able to stop that response. So we asked the question, well, what would be the effect then if we added, what would be the effect on proliferation if we added estrogen in conjunction with BA11-7082? And with the compounds on their own, there's not much happening. But when you add both the NF-kappa B inhibitor and estradiol, we saw a very small, but it was significant, but it wasn't a very large effect on proliferation. So subsequent work then, we were interested in what in the other nuclear receptors as well, and we actually expanded our studies to look at using TACMAN low density arrays, expression of all 48 human nuclear receptors, and on the cell lines, as well as our granulosa cell tumour samples that we had in our tumour bank. And what we observed, in fact, was that there is the second most highly expressed nuclear receptor in these tumours is this receptor, PPAR gamma, peroxisome proliferator activator receptor gamma. And along with its heterodimeric partner, RXR alpha, which is also highly expressed, then we focused our studies a little bit on this particular heterodimer transcription factor. So we looked at expression, and this is work by a PhD student in my laboratory at the time, and she looked at the expression of PPAR gamma in the primary GCT and recurrent GCT, and showed that there's in most tumours, in fact, that this receptor is highly expressed as compared to a normal post-ovary control. This particular receptor has a role in lipid and glucose metabolism, and it's also been shown to promote differentiation of granulosa cells. So again, we this is a conundrum, because why would a differentiative transcription factor be highly expressed in this type of tumour? There are a class of drugs, the thiazolidiones, which are used as anti-diabetic drugs, particularly to treat the metabolic syndrome, that are developed as agonists towards this receptor. And these include these compounds such as triglycerin, rosiglitazone, and pioglitazone. And so when we use these particular compounds in our cells, what we found was, in fact, there was minimal effect on PPI gamma activity, that it didn't induce a PPI gamma transactivation reporter construct. So again, we hypothesized that perhaps it's due to transrepression, and it's transrepression by NF-kappa B. And so I'll show you a little bit of data to show that, whereby when we treated with the compounds on their own, rosiglitazone or retinoic acid, which activate the PPI gamma RXR alpha complex, that we saw minimal effect on cell proliferation. We used a low dose of Bay11-7082, but when we used this in combination with the rosiglitazone and retinoic acid, that we saw this abrogation of proliferation in these cells. Again, to show that it was, this is receptor mediated, when we add an antagonist to PPI gamma in this cocktail, that we are able to somewhat rescue proliferation in those cells. We also showed, using a transactivation assay similarly, that it was only when you have the combination of rosiglitazone, retinoic acid, and the Bay11-7082 compound that you're able to induce this reporter construct. So, so that meant that, well, we hopefully proved from this that, that indeed transrepression by NF-kappa B signaling was responsible. But so just a bit of a summary here, that inhibiting NF-kappa B and activating PPI gamma can induce apoptosis in these cell lines. We still don't fully understand the mechanisms of transrepression, that they still warrant investigating. But the therapy that targets directly NF-kappa B is unlikely due to its critical role in normal physiology. And so our attention is turned to a downstream protein, and it's a protein that's one of the members of the inhibitor of apoptosis protein family, X-linked inhibitor of apoptosis, which modulates NF-kappa B in a feed-forward loop. So this protein, XIAP, is involved in, it's a potent inhibitor of apoptosis for both the intrinsic and extrinsic apoptotic pathway, whereby it binds directly to the caspases that are responsible for inducing apoptotic cell death. And it does so via these three domains, BIR1, 2, and 3 domains. And we know that, my circle's gone in the wrong spot there, but SMAC is an intracellular inhibitor of this protein, XIAP. And SMAC is a mitochondrial protein that is released under genotoxic or stress, and its role is to inhibit XIAP. Just a little bit of info on XIAP. It's a member of this IAP family that potently inhibits caspase activity. It's a potent inhibitor of caspases 3, 7, and 9. And it in fact has a role in granulosa cell biology, whereby FSH has been shown to induce XIAP in rat granulosa cells in vitro, and it does so through the activation of the NF-kappa B pathway. And it's closely correlated, the expression of this protein is closely correlated with granulosa cell proliferation during follicular development and apoptosis during follicular atresia, or when the follicle is not chosen as the dominant follicle and undergoes cell death as a consequence. So Dilys in my lab looked at the expression of XIAP, in a GCT sample here, and indeed in many of the GCT samples we looked at, that there was overexpression of XIAP. There's a little bit of expression of IAP1, another member of the family, but there's no expression of CIAP2 in this in our tumour cohort, and that's compared to the normal postmenopausal ovary. Here we see expression of PPR gamma, which I've shown a little bit expression data before. We also looked at XIAP mRNA levels, but particularly not only is it overexpressed in GCT, but also in the two cell lines, the KGN and the COF cell lines. And so there's high expression of this protein in these cell lines. So given its role in apoptosis, there's actually been a huge amount of interest in evaluating XIAP, and also, as well as the other IAPs, as a therapeutic target for several tumour types. And so small molecule inhibitors are in preclinical development, they've been used in the clinical trials, the various clinical trials worldwide, and these molecules are known as SMAC mimetics. So when we looked at using the SMAC mimetic, what we observed was that when we looked at the NF-kappa B transactivation assay, and this is in KGN cells, that with vehicle treated, this shows the constitutive activity of NF-kappa B. Using the NF-kappa B inhibitor, we see a decrease in this constitutive activity. And when we use the SMAC mimetic compound A, we're able to reduce this NF-kappa B activity in these cells at the same level as what the NF-kappa B inhibitor does. So when we looked at proliferation of our cells, again, this is what we see, a small decrease in proliferation, not significant, with the rosiglitazone or retinoic acid on their own. If you use the SMAC mimetic on its own, compound A on its own, there's a minimal effect on cell proliferation. There's a very small but significant decrease just with the rosiglitazone retinoic acid on cell proliferation. But when you use it in combination with SMAC mimetic, with the SMAC mimetic or IAP inhibition, that we see an abrogation of proliferation. And this corresponded too with an increase in apoptosis. So this is data for both cell lines, whereby it's only the combination of this, of the SMAC mimetic with PPR gamma activation that we see this increase in apoptosis using high content screening. We also have demonstrated in the 3D spheroid model a similar effect, whereby we can grow spheroids very nicely using the KGN cells, and over a period of time with the combination approach, these spheroids are virtually obliterated. And so we're now in the process of doing animal studies, being able to hopefully show an efficacious combination therapy. Also in work that Dilys did, she looked at the invasive capacity of these cells and found in fact that the treatment of these cells with the combination not only delayed the onset of invasion, but also the degree of invasion as well. So it's a fairly exciting result. So as a bit of a summary to that, we know that a combined therapy is a more efficacious approach, whereby we're providing a two-hit approach to treatment, if you like. And the use of the SMAC mimetics will be really useful, because we know that what that will do is it will sensitize the cells, it will lower the apoptotic threshold, and thus sensitizing the cells to lower doses of chemotherapy or other types of therapies that will be used in combination. And it would also has the potential to decrease systemic toxicities that are associated with the higher doses required for a single agent. And these findings may also have broader significance beyond GCT, because we've been able to demonstrate for other cancer types that co-express these particular proteins, that we actually can see some similar results, and that is for epithelial ovarian cancer as well as thyroid cancer. So while the use of PPR gamma agonists may have potential for treating GCT, it's the combination of therapies involving the abrogation of NF-kappa B signaling, and that's through use of SMAC mimetics, it may be of greater efficacy. So just I want to briefly touch on another area that we've been interested in, and that's to look at the genomic events, the mutational landscape, if you like, of what causes the stage 1 tumour to become more advanced or recurrent. So we've done a few studies here looking at whole transcriptome analysis and whole exome analyses. And one of the areas that we've been able to identify is, in fact, is that one of these subsequent hits might involve the TERT promoter mutation. So here, this is the TERT promoter mutation frequency for a whole range of different tumour types that was published by Zahir et al. A couple of years ago, which looked at the prevalence of TERT promoter mutations. And so we know that for quite a number of these tumours, such as gliomas and thyroid cancers, that these are very important. And interestingly, if you look at for ovarian cancer, it's not a feature of, epithelial ovarian cancer. However, when we looked at the prevalence of a particular TERT mutation, promoter mutation in granulosa cell tumours, we found that in fact about 42% of granulosa cell tumours harbour a mutation in the TERT promoter. And in fact, there are 2 hotspots in the TERT promoter that are hotspots for mutations, but we've observed only one of these hotspots is frequently mutated. The other interesting aspect from this work is, in fact, that 30% of our stage 1 tumours harbour this mutation, while 70% of our stage 3 harbour the mutation. And so we're furthering our work to work out whether or not, in fact, these 30% of the women that one should monitor more closely for recurrent disease. There's also been several papers, including ours, where we've looked at trying to understand the genomic landscape of adult GCT. So critical challenge for granulosa cell tumour management is to identify markers that predict late recurrence and or regressive behaviour in order to develop effective targeted therapeutic strategies. We know that from all of these studies, in fact, FOXL2 C134W mutation is pathognomonic for adult GCT. So by far, in all of these studies, greater than 95% of all the adult GCTs harbour the mutation. As I mentioned, there's this high frequency of the TERT promoter mutation that's observed in these tumours, whereby 40% in all, but up to 70% in the advanced. So it's a heterozygous mutation that's present, and so that observation came out, ours and Dr. Pillsworth's paper came out very close together, observing the same frequency of mutation. There's also been identified in other papers too some low frequency mutation in the histone lysine methyltransferase gene, KMTD2, as well as very low frequency mutations in the tumour suppressor gene TP53. Now, that's come out in other studies. In our cohort of well over 20 tumours, we've actually not observed any of these mutations except for one that was in the KMTD2. So specific and putative second hit mutations still remain to be identified, particularly those that are drivers of advanced disease. They need to be identified. And so currently our work is investigating and trying to find whether gene fusions may be involved, which seems to be a feature for other endocrine type tumours, as well as other features such as long non-coding RNAs and so forth. When we turn our attention to juvenile granulosa cell tumours, in fact, the molecular basis of this even rarer type is even more poorly understood. There have been reports of mutations in the G-alpha-S, which leads to the activation of the GSP oncogene. And that's, there's only been one paper really which has outlined that in 2006, whereby they claim that about approximately 30% of juvenile GCT patients have this mutation, and that if you do have that mutation, it leads to a poorer outcome. In our cohort, we haven't actually observed this mutation in our juvenile GCT samples as yet. There's also been a high level, supposedly a high level of AKT mutations in these the juvenile GCT samples, whereby there's an infrared duplication that involves the texture and homology domains that lead to the activation of AKT1. And again, this data has really yet to be replicated by others. So, but these were, both of these were targeted studies. So there has actually been no systematic review of the mutational landscape for GCT, and it's something that we hope to address. So I'll just turn my attention for a few minutes towards the MRFF grant that we were recently successful in being able to get some funding. And this is the title of our MRFF grant submission: Towards a New Era in GCT Research, Patient-Driven Outcomes, Genomics and Therapeutics. And and I'm sorry, this is just a figure that we, just to demonstrate the type of themes that we're hoping to embark on over a 5-year period. And this is a patient or consumer-driven initiative under the MRFF scheme. So what was really important was to have a relationship already built with consumers and patients. And in this circle here, we have several of these, which I'll just touch on briefly in a minute. But in particular, the Gct Survivor Sisters, ROC Inc., the GCTRF, as well as the OCRF, Ovarian Cancer Research Foundation. And the idea with this relationships with these consumer patients and patients is to bridge the gaps in research that currently exist. And so we've built 5 particular themes, and this is for both adult and juvenile GCT, whereby we, and I'll go through what these themes are in a minute, but these are what we see in these particular list here, with the hope to better understand the causes of underlying factors that contribute to the progression and development of this disease. And then hopefully also to improve diagnosis and treatment of GCT, and then the flow on back to our consumer and patient groups. So the partners that we have are this group known as the Gct Survivor Sisters, which is a worldwide moderated members only Facebook group containing over 1,000 patients or survivors. And this group have collated a database with greater than 800 whereby there's greater than 800 GCT patients and survivors have documented their experiences and their journey with granulosa cell tumour. So in this database, we can get a snapshot of what their diagnosis, when their diagnosis was, the advice they were given, the type of management, the clinical management they had to undergo, the types of treatments that they were put on, and the quality of the clinical encounters. And so we've been able to collaborate with this group in order to be able to tease out this database. We also have a very important consumer patient group here, the ROC, known as ROC Inc., or Rare Ovarian Cancer Incorporated, which is a registered charity raising awareness and funds for the juvenile form of the disease. And Tasha Moore is the CEO of this foundation, and is also one of the chief investigators on this grant as our consumer advocate, and is helping us to understand how we can investigate the juvenile form of this disease better. And then also a patient advocate group in New Zealand and Canada known as the Gct Research Foundation, who've also been tremendous in their linkage with patients in those lands. So the themes that are part of this grant are to first of all look at social media and the patient experience. And so we're going to use this Gct Survivor Sisters, what we know is, or we can call real world data, to identify the gaps in patient information, clinical care, health service and support. The second theme is to look beyond FOXL2, looking further at decoding the genomic landscape of GCT, so to identify genomic mutations that lead to the development of adult and also the juvenile forms of this tumour type. So if there's any gynecological oncologists out in the audience, then we'd be very happy to be able to discuss with you how we can gain further access to more patient material in order to continue this work. The third theme is to elucidate the molecular pathogenesis of adult GCT. So even though we understand that that FOXL2 and the TERT promoter are involved, we actually don't understand how they're involved. And so we want to characterize that a little further and hopefully to use that to inform personalized therapeutics. I mentioned at the very start about inhibin assays. We want to develop a more novel diagnostic inhibin assays that is a cost-effective, highly specific and sensitive assay that's based on mass spectrometry. And that's because, in fact, the best form of assay, the alpha inhibin, the total alpha inhibin assay, is no longer available in this country. There are a couple of inferior assays, inhibin A or inhibin B, but they will only detect about 80% of all patients. But if we have a good assay for total inhibin alpha, then we can detect with certainty about 100% of all GCT and monitor all our GCT patients with that. Finally, the last theme is to look at combination therapy, a more precision medicine type approach. So we want to test novel combination drug therapies that includes the SMAC mimetics to specifically target the GCT's pathogenic signaling pathways. So we want to expand on our current work that we're doing there. So some of the other partnerships we've formed in our work with the Ovarian Cancer Research Foundation, Monash Partners, Comprehensive Cancer Consortium, and the Victorian Cancer Agency, who have all, as part of this grant, been very wonderful in being able to provide some PhD scholarship support for us to be able to continue some of these studies as well. So then in conclusions, and why GCT really matter is that also one of the reasons is that rare cancers are often prismatic. And so what we mean by that is that it may help us also down the track to help clarify mechanisms of normal ovarian function, and may also provide insights into other dysfunctions of the ovary. And so I've listed a few of these, and what perhaps the pathogenesis of other sex-caused stromal tumours, premature ovarian failure, polycystic ovarian failure, infertility, which all also involve generally the granulosa cells as well. So perhaps insights from our work on these rare tumour types may also give us some insights into these other dysfunctions. And finally, given the importance of these diseases, insights into our research will, we hope, make a significant impact on women's health, particularly those who have this disease. I'd like to end here then with acknowledgements. Of course, this work has been, there's a whole range of people I'd really like to acknowledge, too many to say in one hit, but particularly my mentor, Professor Peter Fuller, members of my lab, which are listed here and in the picture, Professor John Silk at the WEHI for the provision of compound A and a lot of the insight into how the inhibitor of apoptosis proteins the work and their action, and also linkages here with the patient advocacy groups and Monash Partners and PCCC there. And just my support there, and I think I'll be happy to take some questions. I'm sorry, I'm probably a few minutes over time.
Okay, thank you. Thank you very much, Simon. I hope you can see me now. I can't see me, but I'm going to assume. Yeah, you're nodding.
I can see you.
Excellent. I can't see any questions, Cynthia, but I'm going to kick off anyway and then see what happens. So I've actually got a couple of questions. You've XIP clearly involved. XIP is strongly implicated in this ovarian cancer, but the other IAPs, CIP1 and 2. Now I know you did some immunohistochemistry staining, but I don't think we've actually you excluded that CIPs might play a role, and that does have it is a leading question.
I think when we've looked at expression, mRNA expression as well, we haven't seen expression of IAP1 or IAP2. We have observed though that the use of PPR gamma agonists and the SMAC mimetics actually induces IAP2 expression.
Yeah.
At the message level. So that's an interesting area that we need to explore a little bit. But,
so the leading part of my question is that and is really about the role of TNF. Now I know that TNF has been implicated in ovarian follicle and eruption and lots of things that I don't really understand. But of course, CIBs are very important in regulating TNF signaling. So is there any implication for TNF in this pathway at all?
I think that's an excellent question, and it's one that we're certainly wanting to further explore. I think from our, we've just recently done a RNA-seq experiment to look at the actions of the combination approach, and it would certainly suggest that TNF signaling is important in this process.
Because if I remember correctly, Francis Bulkfield did some studies in ovarian cancer with TNF inhibitors, hoping that it could, but I don't know if she worked on the CGT ones.
I don't think so, no. And I guess when we use exogenous TNF alpha in our system, we actually don't see, other than being able to activate NF-kappa B over mind you, probably a 24-hour, 48-hour period, we don't see much of an effect on the cells. But we've never actually focused on that. But I suppose moving forward, that's certainly an area we need to look at.
I can't see any other questions, but as I said, and I've asked, so I'm gonna enjoy the chance of asking you more questions. So do you think that inhibin, is it playing an important role? Now, and correct me if I'm wrong, but is inhibin a member of the TGF beta family?
Yeah, so it is a member of the TGF beta family, and its role is in fact to suppress FSH in the negative feedback loop of the HPG axis. The interesting thing is it's one of those issues where these tumours are like an FSH primed granulosa cell. However, the overexpression of inhibin would appear to be a consequence of these just being granulosa cells producing a lot of inhibin. We have in the past looked at whether...
Okay, I've got, so I don't want to dominate these questions. Yeah, let other people have a go. I would just mention though that the Drosophila IAP is known to regulate TGF-beta signaling, and it suggests something that we should follow up. Anyway, there's another question
Okay.
from Genevieve Dahl. In terms of the recurrence, what are typical sites, local or distant? Is optimal primary tumor clearance an issue, or is it that these cells metastasize early and then lie dormant until a second event?
That's a terrific question. Generally, I guess that in terms of recurrence, by far the majority are more local rather than distant metastases. It's fairly unusual for them to be to metastasize to anywhere outside the peritoneal region. And I guess for sure, optimal primary tumor clearance is an issue. One of the things that came out of so far our database, looking at the database, was the linkage between recurrence and how the initial tumour was removed. So often, because when women present with this tumour type initially, it's actually not regarded as a tumour, but rather maybe a cyst. And they may go in for a keyhole surgery, the cyst is removed, and unfortunately there may be leakage or rupture of the specimen as it's been taken out, and there seems to be a real connection there with recurrence. And in terms of whether they lie dormant or until a second event, that's something we don't know. The fact that some of these tumours may not reappear for up to 30 years suggests that they can easily lie dormant. Whether they need a second hit or not, we don't know until we can assess the primary tumour and the recurrent tumour.
All right, Simon, I think we've run out of time, but I do want to thank you. That was a very clear, wonderful talk, and it's clear that your work has got this sort of real possibility to translate. So that's really great. And I will just clap on behalf of all the audience and thank you very much for that wonderful seminar.
All right.
Thank you.
Thanks very much. Thanks for having me.